Motivational Program organized by Department of Microbiology, Ramkrishna Mission, Vivekananda Centenary College, Rahura. Today we are very fortunate to have Professor Ramakrishna Sen, Professor and Head Department of Biotechnology, Indian Institute of Technology, IIT Kharagpur, for today's program. We are very grateful to you, sir, for giving us time from your busy schedule for motivating our students. Before we begin, I would like to request our revered Principal Maharaj, Revered Swami Kamalastanandaji Maharaj, to kindly give the welcome address. Hello, Maharaj.
Okay. My chief note. Also, respected Professor Ramakrishna Sen, head and senior professor of Department of Biotechnology, IIT Kharagpur, and my dear colleagues and my dear students of microbiology and other biology department students. Those are present in the YouTube link also, YouTube live streaming link. So we are very joyous moment we are present because with amidst us Professor Ramakrishna Sen, who is very experienced professor in this biotechnology field. He actually started from chemical engineering and now he's doing a lot of good works in this biotechnology field, and from algal biotechnology to biofuel to so many different varied fields. So from him, the students are very much fortunate that you can listen something because he has a very diverse knowledge in this biotechnology. How this application of biotechnology helpful to society and in different fields. He has a lot of work and done lot of patent, lot of projects, crores and crores rupees project he has already has conducted, he has already completed and also running in this Kharagpur IIT. So in this respect, we are very fortunate to having Professor Ramakrishna Sen. And Professor Sen, another his identity, he is our alumnus, especially he is actually the Vidya Mandir, ex-student of Vidya Mandir. So it is our proud privilege that our ex-student also of Ramakrishna Mission, he is actually now in IIT Kharagpur and doing such a great work in the biotechnology. And also in the this Stanford University, this 2% top scientist in all over the India. If you can immediately search, you can get his name. Ranking in the all over global ranking, perhaps within 200, maybe 149 or something. So, so it is very topmost in this challenging biotechnology field. Because biotechnology, many scientists all over world is doing because it is emerging field, and from India he is also a very leading scientist. So with these few words, I express my heartiest regards and welcome to Professor Ramakrishna Sen on behalf of this our college, on behalf of all faculty members, especially you, the students, those are present here and different platform to him for his kind consent for delivering his talk and his motivational talk in this program. So I tell special student, you actually after talk and you interact with Professor Sen wholeheartedly, you learn something because he is actually our ex-student of Ramakrishna Mission Institution, Vidya Mandir. So he very much eagerly wait for your interaction. So with these few words, I do not extend the time because we have already read due to internet problem. I could not start this seminar. So with these few words, I once again express my heartiest regards and welcome to Professor Sen on behalf of the institution. Our thanks to students and other faculty members, those are present here in this beautiful evening. So let us enjoy this beautiful evening. As the thanks to everybody. Thank you.
Okay.
Pranam Maharaj.
Thank you, Maharaj.
Namaskar.
Pranam.
Before we begin, I Before going to Sas talk, I would like to request Professor Subrata Kundu, Assistant Professor, Department of Microbiology, Ramakrishna Mission, Vivekananda Centenary College, Rahura, to introduce our speaker to our audience. Please, sir, you give the introduction speech.
Respected Maharaj, it's my great privilege to introduce Professor Ramkrishna Sen, Professor and Head of the Department of Biotechnology, Indian Institute of Technology, Kharagpur. Professor Sen completed BTech in Chemical Technology from Calcutta University and MTech in Biotechnology from Jadavpur University. He was awarded PhD in Chemical Engineering from Indian Institute of Technology, Madras. He has vast experience over 20 years in academic and industrial research. Professor Sen has published over 230 research articles in diverse high-impact journals such as Bioresource Technology, Renewable and Sustainable Energy Reviews, Drug Discovery Today, Trends in Pharmaceutical Science, Trends in Biotechnology. He has filed total 15 national and international patents. Under his supervision, 24 scholars received PhD degree and 15 scholars are currently pursuing research work. Professor Sen have numerous international collaboration with reputed universities like University of California, Columbia University, University of Pennsylvania. Professor Sen is live member of European Federation of Biotechnology, Asian Federation of Biotechnology, and Indian Institute of Chemical Engineers. Professor Sen has received several awards like Wellcome Trust Institutional Partnership Award, Shastri Indo-Canadian Institute Award, Fulbright Visiting Faculty Award, USA, and Professor Sen have achieved prestigious position within world ranking of 2% Indian scientists published in 2020. Now I am requesting Professor Sen to deliver his valuable lecture to us. Sir, please.
Thank you, Professor Kundu. Pranam Maharaj, and also Pranam to Swaraj Maharaj ji. While bowing at the feet of Thakur, Sri Thakur, Ma and Swamiji, I'm reminded of my days in our commission Vidya Mandira. I stayed in Vivek Bhavan. Our superintendent was Dipankar Maharaj, Sarvaganandaji Maharaj. So I'm just remembering and getting nostalgic. So thank you so much. With sincere gratitude, I start these proceedings. Actually, I prepared a few slides for the students who are studying microbiology. Just for your knowledge, I'm not a microbiologist per se by education. By training, by education, I'm a process technologist. So I'll take you through a journey. But before I start talking about what we have done so far in the areas of energy, environment, water, which has a nexus between these three major areas. We also work in the area of healthcare, and we find healthcare is very much connected to these three. So before I start, I think, I suppose you are doing BSc and also MSc over there, students. Anyway, so let me start this. Can I share my slide here? I just am trying. I'm trying. Can you see the students? The students, can you see first slide?
Yes, sir. See.
Yes, the title is, and it is macro picture of the micro world, genesis to geneth. I hope you understand the genesis of microbiology because you have already studied microbiology in your UG, and also some PG students are here, Maharaj. I'm not sure.
Students of
No, sir, only
botany department, they are also present.
Okay. No issues, no problem. Even I'm in fact a student of microbiology. I have been a student of microbiology so far. It's a fascinating area of study and research. But the more fascinating was for me.
Professor Sen, you make it full screen. Full screen.
Full screen. Okay.
Slide mode. Yes.
Yeah. So for what was more fascinating for me was the drive that the Western world, we usually, for technologies, we look up to them. That is something which needs to be reversed. I hope it will be reversed in another 10 years' time or so. But certain pictures capture our imagination, and we try to figure out what we are doing in this big world of so much of population, so much of demand for energy, demand for water. So the entire humanity is now standing at the crossroad. We are faced with a pandemic, and that has a lot of positives as well. We are going through certain challenges, obviously, but the positives that are more important for us to take and move ahead in our life. So this is hello. Can you
Yes,
hear
yes,
me?
proceed. Yes, proceed.
Can you hear me
Proceed.
and see the slide?
Yes, yes, proceed.
Okay. Just before you, this slide on Kepler 22 system, which shows that the humanity, particularly those who are relentlessly putting their efforts in trying to understand the universe and trying to figure out where the water could be, apart from this Earth, our blue planet, because one day they are some of the scientific stalwarts, they are predicting one day a doomsday will come, or this Earth will not be habitable zone. So as you can see, between our solar system and the Kepler 22 system, there is a habitable zone. And that is what is driving the entire scientific community to search for life, in the extraterrestrial world. So, and in that quest, in that search, already, NASA has reported a moon of Saturn called Enceladus. It has a lot of water. And when there is water, definitely there will be life. People were actually envisaging some life forms in Mars. That's why I hope some of the recent efforts, recent launches by the US and other countries, even mom from our own country, just to figure out whether there could be life on Mars. And I hope your curiosity, if you have already tracked whatever being reported in scientific literature, even newspapers, Mars has Mars gas, MARSH, that means methane, and this methane is produced due to anaerobic action of bacteria. When there are, otherwise methane could not have come over there. So the Curiosity and other, satellites and other, I would say, the search stations, they are engaged in searching for life over Mars. Similarly, we have tried our best to search water on Moon. I think some of the developed countries have already, found water in Moon. Why are you so desperate to find water in other planets or other, satellites of some other planets? It's because we have polluted our environment because of our greed. So much so that the earth may not be a livable place after some time, maybe 100 years, 200 years down the line. And that is why, these are very challenging questions that we are faced with right now. So with this, I'll take you to this. Now, whenever there is water, definitely the smallest form of life would be there. So Earth is blue planet, as you know, and it's also called water planet because 1/3 of it is, 2/3 of is water. Is there any message for me? Okay, no. But truly it is planet of the microbes. And as you know and know better than me about microbiology, but I thought I would introduce you and also learn to you, also learn some of the microbiology, basic microbiology to you. But then, as I understand that I have to talk about our work as well. So I'll take you through these few slides very fast. Okay, as you can see this, science which deals with study of invisible creatures is what is known as microbiology, because it's study of life which is too small to see with the naked eye, unaided eye. Okay. Now, what is microbiology is very much known to you, so I wouldn't repeat this. Microbiology is an interdisciplinary field. It's as you know, there is a paradigm shift of qualitative biology to quantitative biology. And that's how biotechnology has evolved over the years, starting with, say, the production of ethanol to penicillin to recombinant proteins and therapeutic drugs and so on. So as biotechnology has evolved from conventional fermentation technology, and the platform has been the microbiology. And this microbiology now for the last 20, more than that, 30 years, is considered to be a quantitative biology. And there are, other quantitative biology areas that have come up. You might have already come across systems biology, synthetic biology, which are actually, serving as platforms for developing technologies, which are generally known as biotechnologies. Well, so microbiology is an interdisciplinary field, so anybody can study. And you can see the projects, kind of the research work that is going on in the entire globe is purely multidisciplinary now, transdisciplinary. Mechanical engineers are working in the area of biotechnology, in the area of microbiology, biochemistry, immunology, molecular biology, genetic engineering, metabolic engineering, and so on. They are doing this because network, and then computer scientists, they are interested in the network science. So apart from your mobile network, they're also trying to decipher the network architecture of the cells, right? As you know, in humans, you have a neural network. So the artificial neural network has been developed to optimize systems. So these are very interdisciplinary areas. So you should not have a very fixed mindset, my friends, young friends. So that's why I'm saying you just go ahead and venture this beautiful field of microorganisms. As I mentioned, genesis to genet. Genet is something which is just above you, which has come up to the peak point. But still, there's a lot to be studied and discovered, and a lot of inventions need to be done. So innovative minds would work on these interdisciplinary sciences, and definitely at the interface of different sciences, something novel, something new will come up. That is what it has been so far. Nobel Prizes have been given always at the interface between the major disciplines, if you have already carefully noticed. So microorganisms impact, why study microbiology? That you know better than me, because it impacts our life in all possible ways. Like, COVID-19 has impacted in various ways, it has impacted our life, right? Microorganisms impact environment and human health. That they are part of the human environment and the part of the human health as well. In fact, 50% we say, not in terms of weight, but in terms of cell number, 50% of human health is actually microorganisms. And in fact, most of them are friendly microorganisms. These are all God's creations. So they also help us. We have to understand that dynamics as well. I hope you might have come across probiotic organisms. We also work or have worked in that area as well, nutraceuticals. Studying microorganisms provides vital insights into vital life processes. Okay, I'm not going into details because I'll share all the slides that I have. In fact, since I'm not a microbiologist, I have a few students who are from microbiology background. So at the end, I'll acknowledge them. PDFs, postdoctoral fellows who are working with me, the research scholars who are working with me, they have contributed in terms of the slides. So I'll share all the slides that I have got from them. I'll also remember them at the end, acknowledge them at the end. So another reason for studying microbiology is that the study provides insight into life processes in all life forms. Biologists in many different disciplines use ideas from microbiology and use the organisms themselves, right? So that goes on, and actually it helps us to understand more microorganisms, so that the microorganisms constitute the most abundant group of organisms on the planet. That is what I was telling you, 50% of the biological carbon and 90% of the biological nitrogen on Earth are contributed by microbes. Okay, so let me go to the next slides. And human health, microbiota, microorganisms are directly linked to human health. And as we are going through a pandemic, there have been many epidemics earlier. You might have already come across some of the names. The painting depicts the inevitability and inescapability of death for people of all social and economic classes at a time the world was dealing with the deadliest and the most devastating pandemic in human history. That's the bubonic plague. The plague that has been, so now we are going through this, and I was checking the WHO website today, and their numbers are really horrifying. It's more than 1.5 or 1.6 million people who have died, and in India 1.5 lakhs almost. So that is what is bothering all of us while we are staying safe, trying to keep well. We also have learned as to how to take care of others. That is the biggest positive that we have learned in this. And if you keep on doing this, or practicing the habits that we have developed in the last few months, maybe 8 to 9 months, we will remain safer. I tell you, that is how it is in most of the Western worlds, Western countries, that they are habituated to wear masks, they are habituated to keep safe distance, be it pandemic or not. So that is what we are now getting habituated. Hope our health systems also will improve, and the immunity is something at the crossroad. I mean, it sometimes teaches us different things, because people who work on the ground, the villagers, they are more immune to the people who stay in the town, in the not working on the ground. So these are issues definitely, but the practices that we are having right now and the guidelines that we are following definitely will help us improve in terms of our habits. Well, the microorganisms that are very well known, and I hope you know all these, bacteria, protozoa, algae. We work on algae for deriving sustainable, feasible solutions to energy, environment, and water. Then fungi, we also worked on fungi for extracting some or purifying some enzymes. Viruses, definitely our lab did not work. Okay, there are virologists at IIT Kharagpur. If required, I'll request them to give a lecture. Okay, we work on bacteria as well, and these bacteria, as I said, some are friendly bacteria, probiotics, and some are not so friendly, but they produce, metabolites, secondary metabolites, or some pseudo-secondary metabolites, which have immense industrial commercial significance. I'll take you through some of these later on. So as you see the microbes on any surface, it looks like this. We may not be able to see with our naked eyes. So when we see under the microscope, depending on the resolution and depending on the quality and stage of microscope, we can see in different sizes and dimensions. So as we have been using different electron microscopes these days, I hope you might have already come across SEM, TEM, HRTM, and so on. So you can get to see these microorganisms. How do they look like? They are what we call morphologies. So this is how they can look bigger depending on the microscope that you use. Okay, so earlier we used to use just light microscope, very simple microscope, but then compound microscope came, and electron microscope that we have been using. Even we have in our department, it's confocal microscope, which can help us visualize what is happening inside the cell. Where is a particular organelle localized, or if you are injecting a drug, or you are giving something stimulus, where it is going and where it is getting utilized or where it is getting localized, you can get to see on your eyes. So I'll just take you through. I mean, these are. I don't want to use these slides right now because, as I understand, I have to tell you about our work as well. So quickly, this just shows you the morbidity that we are hearing for the last few months, that comorbidity and morbidity and so people with some morbidity, comorbidity issues, they are actually facing awful challenges, health challenges, particularly the senior people. That is where we need to be very, very careful. We have to think about our seniors, our parents, our elderly, kith and kins, relatives, and whoever, wherever we are. We have to be very careful, and we have to take care of them because, once we age, we develop these morbidities, different diseases, hypertension, diabetes, and so on. So, with those complications, this corona or COVID-19 become more dreadful, more dangerous. I hope you have already known from different experiences, different information, source of information. Now, that's why when you think about these aspects, we think they are the villains, right? Because they spoil our food, they cause infections, they cause diseases, and they cause epidemics, pandemics, right? So only 1%, but for your knowledge, for your information, only 1% of all known bacteria cause human diseases. That is, again, silver lining in the cloud. About 4% of all known bacteria cause plant diseases. There are plant pathogens. And in fact, people in our departments are working on plant pathogens as well. And to some people, they actually drive the planet Earth. They have survived billions of years. Can you just imagine how robust their systems could be, that they have survived billions of years? You can find microbes, microorganisms in Antarctica, in Arctics, in everywhere, every place on this earth, right? Like we actually isolated our bacteria, microorganisms from Andaman Nicobar Islands water, because when I started my journey in IIT Kharagpur, I wanted to develop something which is anti-cancer. Though, as I said, I'm a process technologist by training and education. I was not the right person to develop into a drug these molecules, but I worked on, when I did my PhD, I worked on biosurfactants, microbial surfactants. They are amphiphilic molecules with hydrophilic moieties and hydrophobic moieties in their molecular structure. They are called wonder molecules. And if you have a look at the history of microbiology, even recent history, 70s, definitely the name of Dr. Anandamah Chakraborty will come to your mind, right? He developed a recombinant Pseudomonas putica strain which could degrade, aromatic hydrocarbons, recalcitrant hydrocarbons, polyaromatic hydrocarbons, right? So, in fact, his name was nominated for Nobel Prize. We lost him very recently in this year. Dr. Anandamah Chakraborty, I was fortunate enough to have met him. And in fact, his clones, his recombinant proteins, recombinant organisms, bacteria, were the first life forms on Earth to be patented in the year 1982. 1982 is the hallmark year for the biotechnologists or microbiologists, communities, basically the fraternity, because it was the year which yielded the first patent on life form, which has still not been possible in India. Life forms we cannot patent straight away, but definitely it may be a part of the product or part of the process, but we cannot because anything naturally occurring cannot be patented. But what he did, he's actually engineered what he called genetic engineering. He did genetic engineering of, as I said, some Pseudomonas strains, by putting the genes, as you know, from other Pseudomonas strains, which are capable of degrading. So he made a wonder bug harboring the genes of genes that could degrade, that could actually produce the enzymes to degrade. Genes code for proteins, so these enzymes are, as you know, genes. From genes we get genes are transcribed into mRNAs. mRNAs translated into proteins. That's how we get. And we get enzymes. And these enzymes are responsible for degrading the recalcitrant polyaromatic hydrocarbons. So in the year 1982, got the patents, and he was smart enough. He was working in medical school in the University of Illinois. So Urbana-Champaign, and he was smart enough to not to give these strains to anybody else. He actually went for licensing these strains to DAE, Department of Atomic Energy, in the US. And for that, DAE, and then later on, the US government utilized, to my knowledge, I'm not sure, these are very secrets, the military secrets, they utilized some of these strains to degrade petroleum that was in fact spilled on the ocean during the Gulf War. Let's not get into all these, but that's how we remember Dr. Ananda Mohan Chakraborty was a renowned microbiologist and had given so many innovative ideas and products. And that's why today we remember him with all our respect. So some people say microbes, they drive the planet Earth. Bacteria primarily decomposers. They're decomposers, they recycle nutrients back into the environment. They degrade organic materials. Okay, and that's why we use them for bioremediation. I hope you have come across this term, for sewage treatment, solid waste management, and so on. Okay, microbes as cooks. You might have already taken a curd. So curd, actually inside curd you will find lactobacilli, which actually have qualified, they have been established as probiotics. And we work on sporogenous probiotics, which were earlier known as lactobacillus sporogens, but nowadays they are called bacillus coagulans. So we actually have some such strains, and we have developed some nutraceutical formulations based on these probiotics. Probiotics are good for health, for life. Probiotics means for life. There are prebiotics, which are fructooligosaccharides, and some complex sugars, which help these bacteria to grow and which cannot be degraded or digested by us. Well, let's not get into these details. So dahi or curd, cheese, pickles, and other products are produced by bacteria, sometimes yeasts, and so on. Microorganisms per se. Microbes as pharmacists. Even I wish to give you a separate lecture on a different day on recombinant proteins, therapeutic proteins. Today we may not be able to manage time, quality time. In fact, I can request one of my junior colleagues to give a lecture on this. He's the best person to give this lecture on recombinant therapeutic proteins. Microbes, I worked in industry, so there I worked on recombinant therapeutic proteins, including insulin. So there is, and also streptokinase. And I don't know whether you have already come across interleukins, interferons. So those days they were the blockbuster drugs, the blockbuster molecules. So I had the good fortune to work on some of these molecules. Microbes are used to produce antibiotics. It has been known over the years, penicillin from Penicillium notatum, chrysogenum, and so on. Streptomyces species actually produces streptomycin, and so on. So these antibiotics, now, the world actually requires new antibiotics because we are developing resistances to the known antibiotics. The latest one is still a derivative of cephalosporin. Okay. So that's why we are actually lacking new antibiotics, novel antibiotics, which could treat or can treat multi-drug resistant bacteria. That's again a menace, and we can separately talk about that later on. And then comes recombinant therapeutic proteins or prophylactic vaccines. And as far as antibiotic is concerned, I just want to tell you that we also work on antibiotics, anti-cancer agents. We work on lipopeptides. Lipopeptides, they're amphiphilic molecules, also called biosurfactants, and they have shown very promising, very significant anti-cancer activities against breast cancer cell lines and lung cancer cell lines, also colon cancer cell lines. As I said, I am not a cancer biologist, so I worked in collaboration with Professor Mahitosh Mandal at IIT Kharagpur, and he's a cancer biologist, very eminent cancer biologist. And also I worked in collaboration with Professor Arindam Bhattacharya of Baliganj Science College, Calcutta University, on exopolysaccharides as well, anti cancer activities of exopolysaccharides and these lipopeptides. So as we are working towards developing or discovering new antibiotics, I would suggest that some of you also take up this challenge. Take up this challenge to discover new antibiotics, because that's the major demand, the need of the hour. Okay, yeah, we are. There is a prediction. There are two predictions that there will be world war, and that would be on water. And as you know, the dynamics of world war has changed significantly. And I remember the talk by Bill Gates in 2015, who said we don't need nuclear weapons, we don't need armories or arms. A virus is good enough. And he actually predicted that within 3 to 4 years' time, a virus will, kind of disrupt the entire human society. That is what is happening, and he's a visionary, so you could see that. And there may be more future, more dangerous viruses that may be expected to come. As we are playing with our environment, with our water, with our nature, that is why these things are happening. Most probably, we can develop connections through science, but this is what people are thinking, and maybe they are true. So coming to recombinant therapeutic proteins or prophylactic vaccines, I hope we are waiting for vaccines for COVID-19. Hopefully in 2021, the first half, we'll witness some of the vaccines being marketed or being given to the people by the government. That is what we are waiting for. So some prophylactic molecules, prophylactic proteins are also very well known in this business. They are generally called biopharmaceuticals, and they're produced by genetically engineered microbes. Some products are produced by metabolically engineered microbes, because if we can, instead of targeting a single gene for a single gene product, if you target the entire pathway, metabolic pathway, then you can redirect the flux, the carbon flux, the nitrogen flux, and the other nutrient fluxes towards a particular metabolite of your interest, towards a particular product of your interest, so as to enhance its production, so as to make it cost effective, feasible value proposition. So that is why metabolic engineering has evolved from genetic engineering, and systems biology has evolved from metabolic engineering, and synthetic biology has evolved from systems biology. That's how the area is progressing, and you should keep yourself abreast with these developments, new developments, and try to do some research, some kind of studies in this direction. Animal cell culture is the go of the day. Microbial cell culture is not that popular nowadays. Though earlier, all the proteins like insulin was produced from E. coli by Genentech and Eli Lilly. That is also hallmark, as I said, 1982 is the hallmark year for not only Dr. Anandaban Chakraborty's patented microorganisms, Pseudomonas strains, but also for insulin, recombinant human insulin, which saved many lives. It's a life-saving drug, I hope you know. My mother also takes. And the motivation that I got from my own life actually had driven me towards this cancer research. My mother, I have seen her suffer for about 5 to 6 years, and then by the grace of Sri Thakur, Sri Ma, and Swamiji, and the Almighty, my mother could recover, but had diabetes due to chemotherapeutic, I would say, side effects, repercussions. Well, so now she has been going through a lot of other pain and diabetes related diabetic neuropathy and other complications. So she takes human insulin almost on a regular basis. So these are certain cues and certain sources of inspiration that drive us to do something to contribute to the society. I hope you would be inspired by whatever you do, whatever you study, and you are in Ramkrishna mission, and that is the greatest platform. I when I was a student, I used not to realize all this, but as I am aging, growing old, I'm realizing every day the blessings of Thakur Ma and Swamiji. Well, so coming to the topic, genetically engineered microbes, metabolically engineered microbes, we are trying to develop some metabolically engineered algal strains for enhanced lipid synthesis. So these are very specific topics, research topics. I'll maybe take you through some of these slides that we have. It will be difficult to show, I don't know, but some videos also are there. Maybe when I'll be able to come, I wish to come over physically and interact with you. These are not lectures, because I'm also as good or as bad a student as you guys. So I would like to come and meet you and interact with you directly. That time I'll show some videos which will showcase our studies, our research. Microbes as gardeners, you know that some actually fix nitrogen, Azotobacter rhizobium, so they actually fertilize the soil naturally. Microbes as health boosters, I also already mentioned nutraceuticals, probiotic-based nutraceuticals, and there are some examples of microbes which produce E. coli. Again, we actually treat E. coli as some pathogen, but some good E. coli are there. In fact, our gut, our intestine, is inhabited by E. coli and some probiotic organisms. Okay, bifidobacteria and so on, lactobacilli and so on. Okay, so as I mentioned, we work on sporogenous probiotics because if you produce lactobacilli, they usually die. The companies look at the profit, right? So they die while being transported. So you need something which will not die but will serve the same purpose. So Bacillus coagulans actually are sporogenous bacilli. They produce spores, they produce a coat outside, and that's why they can survive all the harsh conditions. And in the supply chain, as they say, the companies, and there is no requirement of refrigeration. So that's why now the market is flooded with such probiotic products, which are mostly lactobacillus sporogens or bacillus. You might find some of our formulations which we developed in Cadila, symbiotic capsules, LMX Fort, Lactocomfort, and so on. I feel happy when I see these still people are using, doctors are prescribing. Well, so these organisms, these probiotics, they produce vitamins. Vitamins B, vitamins, vitamin K, and they produce other probiotic molecules like exopolysaccharides, which help them to colonize in the gut, what we call biofilms. They make biofilms on the gut surface, and they live there. If the population of these friendly bacteria in the intestine, the microbial balance of these friendly bacteria, if it is very high, then our immune system will be very strong. Well, so you can study these aspects later on through your course. And that's why these probiotics have become now. The problem is these probiotics are available freely. They are called over-the-counter products, OTC products, and anybody can go to the medical store and can buy. because they are available in sachet, they are available in capsule formulations, but the level claims are not established. So my challenge after having left Cadila, my challenge or our challenge has been to establish these health claims associated with the probiotics. This is just for your young people, so my students have given this. You may please ignore the picture of bodyguard. So they are bodyguard as microbial flora, which help us maintain our microbial balance in the gut, and that's why our immune system becomes stronger. And that is again, so the probiotics are responsible for that. Very important organisms in our life, friendly organisms. Microbes as cleaners. So this has dual meaning. In fact, microbes produce enzymes which are industrial enzymes. I hope you might have come across amylases, cellulases, lipases, different kinds of lipases, hydrolases. So they are part of the detergent formulations, industrial formulations. They also cleans our clothes, right? Cleans multiple even surfaces and other objects. So microbes as cleaners have dual meaning. So here, if you use whole cells, then you can carry out bioremediation, environmental bioremediation, to clean up pollutants, to clean up polluted sites. You can restore polluted sites. I hope you might have come across Exxon Valdez oil spill. Sometimes these oil spills are engineered, as I mentioned during Gulf War, it was engineered. Some oil spills are just by accidents. Some time ago in Mexico and in Bombay Sea, you might have in Arabian Sea, you might have seen in the newspapers there were oil spills. So how to remove, how to degrade these oil spills? If you put fire, that will kill the aqua animals. It will disturb the ecosystem, and it may cause havoc to other systems as well. That's why it's better to use greener processes, greener methods to cleanse this. So microbial cleaning up, microbe-mediated cleanup of polluted sites and oil spills have been very popular. They have been very, very effective. It takes time, anything, because microbes take time to multiply, produce the enzymes or the products like biosurfactants, biopolymers, etc. So there is another big area which is of my interest as well, microbial enhanced petroleum recovery, wherein we use biosurfactants or biosurfactant producing microorganisms, biopolymers, biogases, biosolvents, bioacids, and so on. Maybe another occasion I will talk about MUR. Xenobiotics are man-made compounds which can be degraded. So as I mentioned, Dr. Anandaman Chakraborty's work, then further evolution in this particular field of work has given us the weapons to remove these pollutants, recalcitrants, persistent pollutants, including polyaromatic hydrocarbons. We have done some work in these areas as well. Microbes as engineers, as I mentioned, human insulin, the biggest discovery. But insulin was the smallest protein to be cloned, overexpressed, and marketed, produced, manufactured, and marketed. But then came the era of insulin-like growth factors, which are bigger than insulin, was not properly expressed by E. coli. So then the era of yeast as host came, right? Yeast. So Saccharomyces cerevisiae, Pichia stipitis, and so on. And then that also was not good enough because the post-translational modifications were necessary for getting active, physiologically active proteins, right? So as the protein size, or rather this therapeutic protein size was getting bigger and bigger, including monoclonal antibodies, which are very popular for diagnostics as well, therapy and diagnostics, okay? Then came the era of cytokines, interleukins, interferons, like the biggest blockbuster drug so far has been erythropoietin, right? EPO, which was produced using CHO cells, not the microbial platform. That's why, as you can see, the two young minds like yours actually joined minds and hands and launched the company Amgen in the US. They were not biotechnologists, they were not genetic engineers. So they actually launched the company with a vision to produce erythropoietin. Erythropoietin is used. I hope you know hematopoiesis. It's used in cancer-related anemia to treat cancer patients which were suffering from inflammation and suffering from cancer-related anemia. EPO became the blockbuster drug in the 90s, late 90s, and also still it is still considered. And then these products were followed by I hope you know EPOGen, which actually flooded the market. It was very, very expensive when it was launched in the US and in Europe, even 3 lakhs per vial in Indian rupees, but now it has come down to maybe 30,000 or so. While working in Kerala, we also launched streptokinase, which is a clot buster. So there are the kinases, right? Streptokinase, staphylokinase, thrombokinase, and so on. There are other kinases which cleave lyse the clots, remove the clots, blood clots. So in ischemic heart attack or in some kind of heart blocks, when we get these clots, these are injected. So they are again life-saving drugs, and they are produced from bacteria. So we used Streptomyces equisimilis to produce this streptokinase. So that is what we need to know how microbes can be beneficial in our life. Human growth hormone, interferons, interleukins. We worked on GM-CSF, granulocyte macrophage colony stimulating factor, granulocyte colony stimulating factor, GCSF, and so on. Okay, so these are therapeutic or prophylactic proteins. Vaccines also, there are recombinant vaccines, and there are recombinant organisms which are taken whole cell sometimes as probiotics with other features like multivitamins and so on. But there are two schools. One is in favor of the GMOs, genetically modified organisms. The another is not. So it depends on the countries who are looking at which perspective. So in some of the European countries, these GMOs are completely no-no, absolutely no-no. Even BT cotton, there had been a debate, right? BT cotton, the Bacillus thuringiensis, it is again genetically engineered cotton so that the insects cannot eat the cotton and cannot destroy. That's why. So there was a big debate. Then BT brinjal, which came into the market, and nowadays you can see improvised vegetables which are mostly genetically engineered. So that's what this fight still goes on, whether genetically modified or not, be or not to be, like kind of a situation. Well, the contribution of microbial biotechnology to the UN's Sustainable Development Goals. I would like to orient you people towards this. I request Maharaj to stop me because I can't see my watch, and okay, maybe mobile I can. Please stop me whenever the time, 5 minutes before the time deadline. So UN's Sustainable Development Goals, I hope you have already come across this term, SDGs, right? So there are 17 goals. I'm just giving you 14, okay? And there are actions that are expected from all the countries, right? So I would share all these slides with you, and let me go to, I think it is 8:36. What's the time? It's up to 9 o'clock, right? Okay, I take it for granted that I can go on till 9 o'clock. I'm not sure. Students, are you there?
Yes, you continue.
Oh, okay. So please stop me 5 minutes before the deadline. Please
Yes,
stop.
yes.
Okay. Pranam Maharaj, Pranam. Well, and the biggest lesson of COVID-19 pandemic, the microbiology literacy, as I was telling you, the healthcare guidelines that we are actually maintaining and abiding by are the biggest lessons, and these are all lessons from microbiology. That's why microbiology is so important, and it's a beautiful area of study and research. Okay, so quickly so never estimate the power of microbes. So I hope in the this is again applied microbiology. That's when you apply microbiology. So instead of going through these, let me share these slides with you. If you have questions, you can send me the questions. Actually, work on as process technologist or as a whatever bioprocess engineer, biochemical engineer, whatever you can say. We work on reactors, different reactor designs. In microbiology, applied microbiology, these also are important for you to know. So I'm just quickly going through these. There are reactors which are called batch reactors, fed batch reactors, continuous reactors, plug flow reactors, airlift or photobioreactors for algae or algal growth, photobioreactors because they are transparent reactors, and maybe some are outdoor, some are put in outdoor conditions. So these are photobioreactors. We have some of these reactors and we grow algae in this. In Bangla, I would say shaula. So microalgae is kind of again micron size shaula. So they actually are rich in lipid or carbohydrate. And if you have lipid rich microalgae, then you can grow the microalgae and extract the lipid, convert that into biodiesel by a process called transesterification reaction. Okay, airlift loop bioreactor, and then tower fermenters, bubble column. So we use some of these reactors and also teach when teaching this particular applied microbiology aspects, biochemical engineering aspects. Well, so I'm not going to cover these, but I will share these slides for your knowledge. Those who are interested, definitely, when I'm not sure when the situation will become normal, you can visit our laboratories and you can get to see the facilities, the infrastructure that we have. Well, and then you can, once you go through these slides, it will be easier for you to understand and follow whatever whatever people will demonstrate. Well, I would definitely arrange for a visit, but not now. This is not the right time. Maybe later part of 2021, and hoping for the best for the vaccines or some other miracle. Upload another slide. Just I don't know how to. Just give me 2 minutes. Stop sharing and then okay. Okay, now I'll share another slide. Just a minute. Just a minute. I'm sorry. Quickly, I'll just go through again some of these slides. This lecture, actually, I had given in ISAR Kolkata 2019. I in a hurry, I could not remove that, though these are old slides, but I think you'd like them. Can you see, students? Okay, I'll put it in.
Yes, sir, we can see, sir.
Okay, thank you. Thank you so much. Now I'll draw your attention to some of our research, not the healthcare research. I wish I could cover that as well, because that's again a big area. I will definitely come back to you whenever it is convenient and whenever Maharaj also find it convenient to call me. I will again come back to you and present our healthcare research, because that we have been pursuing for the last 15 odd years. That's why a lot of data, a lot of information. So I'm not sure how much I would be able to transduce or translate these slides into knowledge in your mind. Let me try. I hope all of you are aware of this Paris Agreement, right? 2015, the UNFCCC, United Nations Framework for Climate Change, its conference. It's a framework convention on climate change. UNFCCC is a big platform for the thought leaders, for the political leaders, government leaders to get together to thrash out issues bothering the humanity, challenging the humanity and also the scientific community. So they take actually resolutions and they try to implement those. I hope you are aware of Kyoto Protocol, which was further, I don't know, strengthened. Hello? Hello? Anybody's saying anything or yeah? Well, so if you are aware of Paris Accord, that also is important for us to understand what we are going or heading towards. If you are able to see this slide, you can see the global warming that is caused due to I hope you might have seen this the wind, the kind of the wheel that rotated, right? So it has actually come to 4 degrees Celsius. Now, in the average global temperature, temperatures have risen 0.75 degrees Celsius in the past 100 years. So how much will they rise in the next 100 years? That is what it is being predicted, right? And if it is even 1 degree more, catastrophic events will be very frequent, devastating lives, devastating civilizations. So forget about 4 degrees Celsius. Now, when it is going to be 1 degree, what is the level of carbon dioxide in the atmosphere? It is about 400 ppm. So what are the scientific challenges, research challenges, technological challenges before us? These are to reduce the contained carbon dioxide level in the atmosphere. How can you do that? Is it possible to target the entire atmosphere? Yeah, some people actually again say that something we can spray from the aeroplanes and then it can absorb or adsorb carbon dioxide. Yeah, that is again some utopian idea, hypothetical, but people can try maybe. But our target is to reduce this carbon dioxide by a process called biological sequestration using algae, photosynthetic microorganisms, right? Cyanobacteria again, and also there's blue-green algae, and green algae are our kind of workhorses towards reducing this carbon dioxide. So we are targeting the thermal power plants. What are the point sources? Cement plants, thermal power plants, they are the major emitters of carbon dioxide. So how can you go ahead and do this? But see, the effect, the impact of this global warming is already visible. Muir Glacier, which is in Alaska, it was looking like this. It was a full glacier, as you can see, in 1941. And what you can see in 2004 is completely melted glacier, right? So all the glaciers are melting. When all the glaciers are melting due to the rise in temperature, I'm not sure, but a part of Antarctica is also melting, right? Sea level is rising. So if this goes on this way, so many cities, including Calcutta, may be flooded one day in maybe 50 years, I'm not sure, 50 to 75 years. How can you stop or reverse this global warming-driven climate change? And climate change is very, very prominent these days. Very often we are getting, cyclones, tornadoes, typhoons, and so on. Natural calamities are very, very common nowadays, right? These are due to erratic climatic behavior, climatic patterns. And remember, we are all water creatures, and microorganisms are part of that, right? And it covers most of the planet, but just 3% is fresh water. So this is, again, we are faced with a challenge. So when our environmental security is threatened, our energy security is threatened because fossil fuels are depleted very fast. Environmental security is threatened because we are going through this global warming phase. The climate change is happening and is real. Then water security is also threatened. And so is our health, so is our food. So when all the securities are threatened, we are going through a phase of insecurities. Then I think there is a clarion call for us to respond. For us, the educated section of the society, for us, to respond to that call and live up to the occasion and deliver. What we can deliver? We can deliver the solutions which are feasible, sustainable, cost-effective solutions, viable. Okay. Now, less than 1% of all fresh water is readily accessible for human use, and most of these fresh water is now polluted. Okay. Due to overpumping, the groundwater in several countries is almost gone. And it has been said, it is also a prediction, we are going to run out of water before we run out of oil. So we are very much worried about oil, right? Whether we can run our vehicles, we can drive our vehicles or not. But before we run out of oil, we may run out of water, and water is life, as you know. For all you know, a water crisis is thus looming large. So we should rise to the occasion, right? The global water security is threatened. So is energy security, so is environmental security, so is health security, and so is food security. And the next, as I was mentioning some time ago, next world war is predicted to be on water. This is how it is going to be. Say a Bisleri bottle now costs about 20 bucks, 20 rupees a liter maybe. Okay, and if you are seen with a Bisleri bottle in your hand, 20 years down the line, it may be snatched away. 30 years down the line, you may be attacked, and one may be killed just to carry some such water. It would be a fancy because other people are starved or thirsty of water, right? So with this background, I'll quickly take you through this journey, which is climate change-driven, bio-inspired journey through some contemporary research challenges of energy, environment, and water. Well, so as I was mentioning, that humanity is at the crossroad. There are so many challenges that we are faced with. Quest for sustainable solutions to these global challenges has to be on, and we must be on our job to deliver the solutions. What are the solutions? We, in our own way, in our own small way, we are trying to understand, trying to figure out the global scenarios and develop biorefineries. As you know, petroleum refineries, they deliver, one is there in Haldia. It actually processes crude oil and delivers multiple products, right? In the fractionation column, right? We get LPG at the top, then naphtha, then we get kerosene, then petrol, what you call gasoline, then diesel oil, and so on. And then finally, at the end, what you call bottom part of it is wax. And then farther bottom you have asphalt, bitumen, and so on, with which we lay the roads, right? So there are multiple products that one can derive by processing crude oil, crude petroleum. Similarly, we asked ourselves, can we process biomass in the same way to derive multiple products? So you might have already come across biomass-based biofuels, bioethanol, biodiesel, biobutanol, and so on. They are considered to be, alternative to fossil fuels, because fossil fuels are getting depleted day by day and very, very rapidly. Though there are geopolitical reasons to believe that some countries may have very high, storage of reserve of fossil fuels due to geopolitical imbalance, hegemony. But the rest of the world is actually going to suffer from this crisis, energy crisis. So can we develop alternatives to this energy for fossil fuel energy, petroleum or coal? So with that kind of vision, we started working, and since 2009, we have been working to develop microalgal, microbial biorefineries with a holistic and viable, with a holistic vision, and for developing viable model for energy, environment, and water. Because there is a nexus. They are all connected to each other. If you disturb one, the other one is automatically disturbed. There is a chain. And in this chain, you find food and health as well. Okay? So I just again remind you of the 17 Sustainable Development Goals. So those who will be inspired by these presentations, the two presentations, I hope will remember and try to achieve in your life. It takes time, but it takes efforts and courage. dedication, devotion, okay, to achieve at least one of these 17 goals. Okay, and we are all talking about nowadays circular economy. There is nothing like waste, W-A-S-T-E. Waste cannot be wasted. Nothing like, so every waste can be converted into wealth further. So it's circular economy. So you process something, get some products, get some wastes, convert that waste into wealth. So this circularly it will go on, and it's called circular carbon economy. You'll get to know better when I interact with you physically. Well, so with that vision, with that kind of intention, with that kind of drive, we started our research. We have been doing it for the last 11 years or so on in these particular areas, energy, water, energy, environment, and water. While keeping in mind the technological challenges, the energy input, water input, land use change, real estate is a big business. So that will be a competition for these technologies. Greenhouse gas emissions. So are we actually doing something and emitting more greenhouse gases than the power plants? That is the question. What is the scale that we can operate? How much, or what is the quality of labor? And while thinking or developing such biorefineries, are we really considering the sustainability issues? What are the sustainability issues around the feedstock, the biomass itself? You might have come across Jatropha, might have known other vegetable oils, which are some of are edible, some are non-edible, some of are non-conventional non-edible. You might have come across biomass, lignocellulosic biomass, right, which are the feedstocks, but are they sustainable? I mean, if you want to develop biofuel solutions, can you get these energy crops grown, cultivated in lands? Where is the land available? Where is the water available to cultivate, to irrigate? So these are the questions. So I request all my students here and also in IIT to think any research problem, any scientific problem that comes to your mind holistically. I can just have a window approach, solve something very temporary solution I can bring in. But if you think holistically in terms of feedstock sustainability, in terms of environmental sustainability, in terms of economic sustainability, in terms of social sustainability, in terms of political sustainability, geopolitical sustainability, are these really sustainable? Sustainable, the term actually means that we are doing something for the next few generations without compromising with the natural resources for the next generation. So we should do. I'm just in a hurry, so I'm going. So in this you can see we operate these microbial and microalgal biorefineries and derive multiple products. You might have already seen microbial surfactants for cosmetics, food, pharma, MUR. As I said, some of our anti-cancer agents, some of our strong antibiotics, broad spectrum antibiotics. We also derive biofuel solutions, we derive therapeutic solutions, we derive probiotic solutions, nutraceuticals, we derive enzymes, we derive pigments, and other antioxidants, right? Some other healthcare molecules. Some are patented, some, but I'm very bad at business, so I could not or haven't been able to leverage the patents and convert them into some economic proposition. I have not been able to do that. Now our footprints, I'll just go through very quickly. This is how we started. In 2009, I somehow, by the grace of again Sri Thakur Ma and Swamiji, Sri Ma and Swamiji, I got connected to this group, and it was by the blessings of two stalwarts gentlemen. One is Dr. P.K. Sinha, who is an alumnus of IIT Kharagpur, so I remember him, acknowledge him. He has actually given an endowment fund to our institute, and an bioenergy center has been developed with his fund. It's called P.K. Sinha Center for Bioenergy. I was initially the coordinator of that center, I happen to be. And Dr. or Professor Chris Somerville, who is the director of the Energy Biosciences Institute, University of California, UC Berkeley, California, Berkeley. So these two gentlemen actually have taught me lessons which have helped me to grow and think all these in a very holistic fashion. So this is how I got to meet all the world experts, world leaders in the area of energy, environment, and water in Portugal. It was long back in May 19, 2009, and that actually inspired me. As I said, inspired me and my group to go through some of this journey. So let's not consider all these impact factors and all these. It's now 30 point odd, 2019, but these we put for our institute presentations and put for funding agency presentations. So please ignore these things. Well, this is about NUR, which I'll talk about or talk on later on. Petroleum recovery. Can microbes be sustainably used to recover petroleum, more and more petroleum? Definitely, when we recover more and more petroleum, it will be at the cost of environment. But we have to take care of that as well. Can the processing in petroleum refineries in companies like ONGC and Oil India or Shell, Chevron, or BP, British Petroleum, and so on, be green based on green methods, bio-inspired methods? Then we can, right? And then we figured out that actually the microbial surfactants can do the wonders, which actually were discovered by, or rather made popular by Dr. Ananda Mohan Chakrabarty. So we have been working on these molecules, and I did my PhD also on these microbial surfactants in long ago, 1993-97, in IIT Madras. Well, so we have been working in this area. I'll discuss these things later on, but biofuels I'll bring in. So once we say that we need alternatives to petroleum or fossil fuels, then we give the reasons that world is running out of oil. But remember that we are actually running out of water before we're running out of oil. And there was a declaration even by The Economist and the Times magazine that there's the end of the oil age, and it happened in the year 2008 when the oil production peaked. But we were not aware of the geopolitical scenarios. So then, Gulf War was one of the reasons. And then there were other issues. I don't want to get into all these. So that actually drove us towards more of renewable energy process development, product development. So we started mapping the world scenario, the global scenario, as to how lignocellulosic bioethanol could be an alternative to this. And this is how the bioethanol can be produced from lignocellulosic biomass, which is not in competition with food crop. I hope you are aware of food versus fuel debates. We cannot take any biomass which has food value. So we cannot take cornstarch, we cannot take maize, we cannot take sugarcane, and so on, right? Because they are food stuff for us. So can we take hardwood, softwood, leaves, corn stova, bagasse, and so on, and maybe straw? But straw again, there are societal issues in developing countries. People make shelters out of straws. Straws are good fodders for the animals, and so on. These are the social issues that we should be aware of. So we targeted the last one, that is the rice husk, which is the tough nut to crack, and we took that challenge and went through these process development exercises. Soumita Banerjee did her thesis on this. She developed wet air oxidation and then improvised this process further to alkaline peroxide assisted wet air oxidation, and could deconstruct the biomass to release cellulose and hemicellulose, and could separate lignin out of it. Cellulose and hemicellulose were further sacrificed, what we call enzymatic saccharification, by using cellulases that we got from an industry out of Nagpur. Well, so we converted enzymatically that cellulose, hemicellulose produced sugars. These are not sugars from corn, neither from corn nor from sugar cane. So we are not encroaching upon the food part of it. So we developed a conceptual process, integrated process, and which can be translated into an industrial process easily. This is what later on I'll So basically, we could figure out that separate hydrolysis and fermentation would work better for converting lignocellulosic biomass, as I said, the rice husk, into bioethanol. Bioethanol was used to be considered as an alternative to petrol, but it can never be because it has oxygen in it. It has less energy density, less calorific value. So it can be used as supplement. That is what government of India has mandated. From 2017, it wanted to start, blending the petrol, gasoline with bioethanol, but supply is so meager that is still not implemented. Now, we got a patent out of this that is from another oil. I will skip these slides. Well, and this is something again a God connection, which brought us to the world map on biodiesel. Okay, suddenly an US group visited us. As you can see, Jeff Thompson is there beside me, and Dr. Ashok Desarkar, who happened to be our alumnus of IIT Kharagpur. He has a big business in Houston, in the US, and Jeff is basically consultant. So they visited my lab, and we are just doing very simple method. We are implementing very simple method for the production of biodiesel, and they were very much impressed with that. It was a BTP project, BTech project. As you can also do some projects like that. Can you imagine a UG student developed this, and this caught the imagination, captured the imagination, and captured the eyes attention of this US company, and they funded our project, and we developed, on the right hand side you can see, we developed a process which was further patented. It is an integrated reactor system to produce biodiesel cost effectively and biocatalytically, enzymatically. So that's a green method, green process that we developed for the production of biodiesel from any oil. It is irrespective of any oil source. We use different oils and produce biodiesel. These are biodiesel products. Before you tested the biodiesel products in diesel engine, we found favorable emissions and other characteristics, fuel characteristics and properties, and that is why they liked it. And we actually filed a patent, continuous biocatalytic production, as you can see on the top patent, and that patent was taken by the company. So that was a successful venture within 18 months. So that was something. So I again owe all the hard work to Soham Chattopadhyay, who worked, Professor P. Das, who also was kind of a guiding figure. And then we understood that lignocellulosic biomass cannot be a sustainable feedstock. Where will you get the land from? Where will you get the water from? And where will you be able to produce this sustainably? Because these plants will take its own course, time to give you biomass, right? So that's why we switched over from lignocellulosic biomass to algae. In the US, they say algae. But we say algae. So I will use the word algae. So as you can see, we can use sunlight, we can use wastewater, and we can use CO2 being emitted by the point emitters, thermal power plants. And these are so far these are free, right? I'm not sure. Wastewater will not be free after maybe 4 or 5 years from now. But we got some wastewaters from industry, Haldia, some industries from Diamond Harbour. Okay, so we got wastewaters. We got wastewaters from our municipality. And we cannot take carbon dioxide or flue gas from this thermal power plant. It's a huge structure, right? But we got the heat transfer data from them, designed our own flue gas generator. I'll show you the picture, and developed these. So what we are deriving out of this, if you consider this microalgal biorefinery with algae as feedstock, then biofuel you can get, clean water, you are processing wastewater, producing clean water, may not be drinkable, potable water, but processed water. You can produce biofertilizers, you can produce animal feed, fish feed, you can produce pigments, which are very good antioxidants, and you can produce even PUFA, MUFA, which are again very good healthcare molecules, nutraceuticals. So with that vision and value proposition, we had gone ahead and developed an algal biorefinery, developed nutraceuticals products out of algae, treated wastewater, so bioremediated wastewater, produced value in terms of fuels, water, in terms of, and food, feed, biopolymers, pigments, biofertilizers, and so on, multiple products. That's why we are calling it a biorefinery, algal biorefinery. At the same time, you can see carbon capture and storage. We are actually capturing carbon dioxide, converting that carbon dioxide into biomass. And we are also having a scheme to convert the rest of the carbon dioxide, which is not captured by the biomass, into methanol, into some other products. So this is the pathway. I'll quickly again take you through this. We started with some marine microalgal cultures. These cultures were not gifted, but we are in a collaboration, and a big collaboration is called NIMITLI program of CSIR, New Millennium Technology Leadership Initiative program. So I was in charge, or rather I was made responsible for carrying out the downstream processing part of it. And the algal cultivation was done by CSIR, CSM, CRI. It was a big project among 9 different institutions. We were one of them. And as you can see, with the microalgal culture on a plate, we actually maintained that culture, developed the facility, 3-liter microbial culture reactor converted into photobioreactor, then 30-liter reactor. Okay, we designed and developed. And then we actually grew this particular culture in the open raceway pond. It's called raceway pond. It's an open cultivation system because these, as you scale it up, these closed photobioreactors will be very, very expensive because that would require control of, sophisticated control of temperature, pressure, pH, agitation, and so on, aeration, and so on. So that's why we cannot afford expensive reactors for the production of biofuels, which are very large volume, low value products. We cannot produce biofuels like therapeutic proteins and sell at 3,000 rupees a liter. We have to produce at a very cheap, at a very inexpensive price, say 20 to 30 rupees a liter, and sell it at 60 rupees a liter. Then people will take, right? Market dynamics. So then we also found that harvesting of such microalgal biomass from such a huge amount of water pool was a big challenge. So we developed a dewatering unit, which is patented again. This is very interesting. Is a conveyor belt system, and there is an adsorbent that is underneath, as you can see, underneath the biomass, and we could actually dewater. I'll quickly go through, and then as you can see, the biomass grown in open raceway pond, and then lipid was extracted from this biomass, which was converted into biodiesel process called transesterification, which was catalyzed chemically as well as biocatalytically. We used lipases again, and we actually characterized the products which are called FAME, fatty acid methyl esters. And this is the flue gas generator from which we got flue gas. We could scrub off SOx and NOx, and got the gas sparged into the, again, small scale raceway ponds, which are there on the right hand side, to my right hand side, and sparged that flue gas and grew microalgae, extracted lipid for biodiesel, extracted carbohydrate for bioethanol. Okay, that's how it is. So this is the entire process chain that we developed. Some of these part are patented, protected, okay, but I have not been able to translate that into business. Okay, so we also used some wastewater, poultry waste, and some other solid waste, mixed them, and developed green integrated process for the mitigation of municipal and industrial liquid and solid waste for the enhanced biomass production. So this is again the pathway that I have shown, and on the left hand side you can see, to my left hand side, you can see the flue gas generator, which is just it's just duplicate of what we see in a thermal power plant. Okay, we replicated that. We developed a smart green reusable nanocomposite based cell harvesting and disrupting system. That is also a major scientific challenge we could solve. That is published in ACS Sustainable Chemistry and Engineering. So our engineering vision is like this. We would like to operate in tandem in collaboration with the thermal power plant. We'll actually try to produce more biomass, reduce coal consumption, operate this carbon recycle process, and we'll try to achieve CCR, carbon capture and recycle. We also have developed yeast biorefinery lab scale technology development. There are oleaginous yeast, so we have derived multiple products, bioemulsifier, biodiesel, biomass itself, which can be converted into bio-oil by hydrothermal liquefaction or pyrolysis. And in this process, we got some water, which is purified, and we have developed biomass-based water purification system. As you can see, we have got the US patent on this. Anyway, I don't know whether it will work. I'm not sure if it works. Can you see the video is working?
Yes. Is there any sound?
Can you see the video?
Is there any sound?
No, Maharaj. This will show something which I actually have tried so far.
No,
Just wait for a few seconds. Wait for a few seconds. This is what I have tried for the last maybe 2 hours. I'm not sure. By your blessings, Maharaj, this is what I have tried. And in just one picture, students will be able to understand what I have tried to do. I have tried to draw a picture in your mind. I hope that would be everlasting. And this particular picture will hopefully will be there in your memory for the entire life. With that, I thank you very much. And I acknowledge, as I said, Chris Somerville, Dr. P.K. Sinha, Sri Sri Thakur, Sri Ma, and of course, the one infinite, pure and holy, beyond thought, beyond qualities, I bow down to thee, Swamiji. Thank you very much. Thank you very much. Thank you very much. Pranam, pranam, pranam.
Thank you, sir, for your wonderful,
I'm sorry for
encouraging
taking
speech.
longer time.
I think all of our students are encouraged.
Thank you very much. Thank you.
Thank
Thank you
you,
for
sir.
the opportunity and honor. Pranam to everyone. Everyone.
Now I request our student, if you have any queries, if you can ask, sir, you can free to ask any questions.
Yes, yes, please.
Hello, sir. Am I audible?
Yes, yes, please go ahead.
Yeah, thank you, sir. I am Rishabh Kaur, and sir, I'm very much interesting about the field of bacteriology, and
Right.
I will also want to do research in the field of bacteriology in future. And sir, I want some guidelines from you about how could I design my pathway from the current BSc to that goal of research in future in the field of bacteriology.
Right, right. See, microbiology, applied microbiology, or bioprocess, and then these are all connected to molecular biology, genetic engineering, metabolic engineering, systems biology, synthetic biology. There is enormous scope for research. So all you have to do is to orient yourself towards the needs, the demands of the present civilization, right? As I was telling you, the journey that we have undertaken, the journey that we have undergone, actually are all driven by some of the cues, some of the inspirations that I have already acknowledged them, and I forgot to acknowledge my students. I acknowledge thankfully and gratefully Dr. Chinmay Hajra, who is a PDF postdoctoral fellow, Dr. Anirban Dasgupta, who is a PDF postdoctoral fellow working with me, and Mr. Ramit Mitra, who is a second year PhD scholar working with me. They all have contributed to the earlier PPT. And the second one actually are all our research slides contributed by many students. Well, so to coming to your question, so first thing is, what inspires you? There are various facets and aspects of microbiology or applied microbiology or biotechnology. What actually inspires you, your mind? What ignites the fire within you? So just try to figure out this, and accordingly orient towards that. And I'm sure the college, it's has all the strength and capabilities and infrastructure, and as well as we are there. We are part of you, right? We are not different. I'm just as one of you, as I mentioned, as good, as bad as you, all of you. So we are there as a peripheral support system. So when your mind is ignited with an idea, if it is kindled, you got this, you've got the spark, then you pursue that idea. That's what Swamiji also has said. The dream, that's why you have to chase the dream and leave that. Whatever idea you have, you think about that, leave with that idea. Okay, so that is what is very important. And in that journey, if you need any help from our side, you are always welcome. But as you know, you see the pathway from BSc to MSc and then research. That's how you have to. You cannot escape this ladder or the steps, right? So only thing is from now onwards, if if you think that experimental ideas are bringing some joy to your mind, it's about joy. You have to enjoy the journey. So or theoretical ideas depends. Nowadays there is no boundary. Bioinformatics has evolved over the years, and in fact experimental merged with theoretical. That's why we are seeing systems biology, synthetic biology, designer organisms. One day you will find designer organisms which will do the job even of a robot. Designer organisms which will deliver the products that you want at a very high concentration at a particular place. Okay, so these are going to come, I'm sure. Well, so as I have shown you some of the dreams and some of the out of the earth, out of the box ideas, if one or two or even some other ideas give spark in your mind, pursue that from now on. And if it is experimental, ask your professors to help you set up some experiments towards achieving that bigger picture, the bigger goal. Well, and during that journey, if you need help from our side, please let me know. The situation is not normal, so you may not be able to come now, but maybe after a year or so, you may come and implement the idea to the extent that you want. Is it okay?
Thank you.
I'm not sure.
Thank you, sir. Thank you so much, sir.
Okay, thank you. Thank you. Any other question?
Hello, sir.
Yes, hello.
I am Anubhav.
Anubhav, yes.
You have mentioned about projects in your talk. So, as an undergrad student, we often find difficulty to get such projects. So, is there any opportunity for us to do such projects in
Yes.
IIT KGP?
Yes. I mean, I wouldn't say projects because that way, every system has its own dynamics. So project takes about 3 months to 6 months, right? So for that, an institution has to sign an MOU with IIT if some of the students do projects here, which are not really encouraged nowadays because of infrastructural limitations. Number one, and number two, it's not about your college or my college, RKM Vidya Mandir. It's about, but that's how it is. Some systems which grew, mushroomed in the last 10-15 years, which actually tried to take undue advantage of the IIT system. I hope you are mature enough to read my signals, right? Some of the colleges which have mushroomed in the last 15-20 years, even in West Bengal and neighboring areas, they have tried to take undue advantage of the certificates, some projects. So these projects have been stopped from 2010 onwards by a Senate approved rule. So, but that doesn't stop us, from bringing you here. So all we can do is during summer, 2 months we get, and during winter we have summer vacation, winter vacation we get about a month. So in total we get 3 months. So in summer, you can come as summer interns. Internship you can do in IIT. So once you are familiar with the system, and then you can come back in the winter for 1 month. Well, and then you can come back again in the summer for 2 months. That's how we are supporting. But since a rule is a rule, so that was not for the colleges like your Ramkrishna Mission Centenary College or any Ramakrishna Mission college or school, but these rules were made for. But you cannot discriminate. These rules were made for the other colleges which were, as I was taking undue advantage of the IIT system. I hope you got my signal and my message. Whenever you think you can do project, you just call it internship. The name would be different, but you will do a project in our labs. Well, depending on your choice. So you can write to me. I will guide you to the particular lab or to the particular professor of your interest, or if of research of your area of your interest. So accordingly, I can connect you to the professors. Or if you are interested in some of these areas like healthcare, but up to product process and product development, because we produce molecules and purify them. So like as I mentioned, anti-cancer agent, actually we purified up to 96, 97% purity level and had given those molecules to Dr. Mayutosh Mandal and Dr. Arindam Bhattacharya, who are cancer biologists. So they studied. In fact, we worked in collaboration with Peerless Hospital. There is Dr. Shatadal Das, a very good microbiologist, who helped us to decipher the broad spectrum antibiotic properties of our molecules. So these molecules are new molecules, as I was telling you. So if you are inspired by some of these products and processes, then you are welcome to work in our lab. Our lab is known as BBDL, Bioprocess and Bioproduct Development Laboratory. Is that okay?
Yes, sir.
I'm sorry whether you are missing your dinner. I'm not sorry. I'm really sorry about that. If you are missing your dinner. Any more questions?
Any other question you have?
question থাকলে জিজ্ঞাসা করুন।
I tell you, you're welcome to visit our laboratory or department anytime. But as I said, we have to wait because of safety for all concerned. We may have to wait for a year or so, and then I'm not sure those who are in third year, if they are going to do MSc in the same college, then you are always welcome to visit us. But now our labs are not functioning. For the last almost 8 months, our labs are not functioning. Research students are not here in the campus. So that is a big concern for us. We are just stalled. We cannot move forward. That is what is. So then, those who are theoretically strong, they can do just with a laptop or with a desktop. You can do a lot of bioinformatic analysis, which say network development, molecular networks, molecular switches. So those come under systems biology, synthetic biology. Okay, any other question? If not, then let me thank and express my gratitude, my pranam to Maharaj. Swaraj Maharaj, I have also seen my pranam to you, a pranam to all professors. I bow down, as I say, bow down to thee.
Professor Sen, wait, just vote of thanks maybe.
Okay,
Yes, sir. Yes, sir.
Please give vote of
Okay,
thanks.
Yes, sir. Okay.
Please
okay.
tender
So
vote of thanks.
with this, we come to the concluding moment of today's program. On behalf of our college, on behalf of our all staff members, all students, I extend my really heartfelt gratitude to sir, who spread his busiest time from his busiest schedule to grace the occasion. I also thankful to him for a wonderful talk and interaction. And today we had an opportunity to hear his thoughts, and this will surely be going to encourage us for our future progress. and we will hope that he will definitely enlighten us again. And after the pandemic is over, we invite you, sir, to please visit to come our college, and
My
we will
pleasure. My
enlighten
pleasure.
you, sir. So
My
thank you,
pleasure.
sir. Thank you.
Thank you. প্রণাম প্রণাম। সবাই ভালো থাকবেন।
হ্যাঁ,
আমি
নমস্কার,
প্রার্থনা
ভালো
করি
থাকুন।
সবাই ভালো থাকবেন।
আপনারও ডিনারের দেরি হয়ে গেল, 9:30 বাজলো।
না 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 না, আমার কোনো দিনের দেরি হয়নি, আমি পরেই খাই, আমাদের 10টা হয়ে যায়। আপনাদের
যে
শুধু
ওয়ান্ডারফুল
ডিনারে আমার
কাজ।
খুবই খারাপ লাগছে এখন, এটা আমি আমি অত্যন্ত দুঃখিত ডিনারের দেরি হওয়ার জন্য।
না না, ওদের দেরি কেন হবে, ঠিকই আছে,
আচ্ছা
কোনো
আচ্ছা।
অসুবিধা নেই। কিন্তু যা যা খাবার দিল, মানের মানে মানে ইন্টেলেকচুয়াল ফুড যা দিয়েছেন, এটা আবার তো যথেষ্ট। এটা যে সমস্ত ফিল্ডে আপনি কাজ দেখিয়েছেন, এটা তো মানে সো মেনি থট প্রভোকিং আর কি।
আমি আরো মহারাজ, আরো যখন আপনাদের শিডিউলে আমার টক ইনভলভ করা যাবে, ইনক্লুড করা যাবে, আমাকে জানাবেন। আমি আরো কিছু তৈরি, কারণ এই সময়টা কি হলো, আমাদের এই 26th আমাদের সেমেস্টার শেষ হলো, সো মার্কিং গ্রেডিং অনেক খুব হেক্টিক টাইম গেল আর কি। হ্যাঁ, তো আমি ঠিক গুছিয়ে উঠতে পারিনি, তবে এবার আমি ঠিক একদম 1.5 ঘন্টার মতো বানাবো। আমি খুব ভালো করে হেলথ কেয়ারটা বোঝাতে চাই ওদের।
হ্যাঁ নিশ্চয়ই, এরপর আবার একটা হেলথ কেয়ার বলবো। আর আপনি যে বলেছেন ওই আপনার জুনিয়র দিয়ে ওই এটা করাবেন, ওই কি একটা বলছেন তো, হ্যাঁ ইয়েস ইয়েস ইয়েস, হ্যাঁ ঠিক,
রিকম্বিনেন্ট প্রোটিন, ঠিক ঠিক,
রিকমেন্ড প্রোটিন, হ্যাঁ ঠিক।
হ্যাঁ আমি আমি আমার জুনিয়রকে বলে ওটা একদিন ব্যবস্থা করব। আপনি আমাকে ডেটটা জানাবেন, প্রয়োজন ডেট।
ঠিক ঠিক, ওটা খুব ভালো ফিল্ড, একটা রিকম্বিনেন্ট প্রোটিন খুব ভালো।
ঠিক
ঠিক, যাক, ভালো থাকুন, শরীর ভালো রাখুন, ঠাকুর মাস্টার যে
প্রণাম
কাছে
নেবেন মহারাজ, স্বরাজ মহারাজ প্রণাম নেবেন।
নমস্কার, প্রণাম নেবেন, নমস্কার।
প্রণাম প্রণাম প্রণাম।
ভালো থাকবেন, ভালো থাকবেন।
অনেক অনেক ধন্যবাদ, প্রণাম আমাকে সুযোগ দেওয়ার জন্য, আমাকে সুযোগ
না
দেওয়ার
না,
জন্য।
খুব ভালো থাকুন।
আমি বিদ্যা মন্দিরে এসেছিলাম এই বছরের গোড়াতেই, ডিসেম্বরে এসেছিলাম মঠে এবং বিদ্যা মন্দিরে, কারণ আমার বন্ধুরা যারা নিউ ইয়র্কে রয়েছেন ওরা ভিজিট করছিল, ওরা ইনফ্যাক্ট ছিল গেস্ট হাউসে। তাই জানুয়ারি মাসে এসেছিলাম, ডিসেম্বরেও একবার এসেছিলাম, কিন্তু জানুয়ারি মাসে আমরা কামারপুকুর, বেলুর মঠ হয়ে কামারপুকুর, জায়রামবাটি হয়ে খড়গপুরে আসি। কিন্তু আমি জানি না আবার কখন যেতে পারবো, ইচ্ছে আছে তাড়াতাড়ি যাওয়ার। হ্যাঁ
ঠিক ঠিক।
লাস্ট অ্যালামনি মিটেও আমি গেছি।
হ্যাঁ, এই বছর তো মনে হবে না, মনে হয়, কারণ
এই বছর তো সম্ভব নয়, লাস্ট অ্যালামনি মিটেও গেছি।
ঠিক ঠিক, এই আর কি, এই ভালো থাকুন, গিয়ে এতে করুন।
ভালো থাকবেন মহারাজ, প্রণাম।
ভালো।
মিটিং, হ্যাঁ।